much for nice introduction and invitation, and thank you for everyone for coming. Um, I am a not developer, but I am a end user. So today I would like to discuss about how to use the MPS for assessing the drug-induced toxicity as a from the toxicology standpoint. So rationale why we need the MPS to capture these kidney toxicity is that from the in-house our experience, our in-house molecule experience the dose-limiting toxicity with the proximal tubular degeneration in both rat and dog studies. And we don't know the MOT mechanism of toxicity, so that's why we of course don't know the translatability between the preclinical species and the human. And in addition to these molecules, there are many emerging new modalities such as ASO, for instance, which expected to see some sort of nephrotoxicity as published, and other molecules. So we really anyway need to have kind of capability to capture the nephrotoxicity of the compounds. So our goal is, and expectation is to modeling the 3D MPS, which is expected to see the physiologically more relevant to the in vivo situation of a human organ. And so that we expected to see the enhanced sensitivity to these drugs, and eventually applied to the strategy by using the readout as a readout for the future molecule prediction of the nephrotoxicity. So briefly touch on about the kidney, maybe not necessary to talk about, but basically the kidney role of the kidney is actually the excretion of the waste, including drugs. There are two main routes. One is glomerular filtration. During that part, there is a chance to get injured for these glomeruli by the drugs. Second is proximal tubular excretion, directly from the blood circulation to the urinary excretion. Both are basically depending on these transporter-dependent excretions. But not only the excretion of the drug and the waste, but also in charge of the reabsorption of the required substance, for instance, nutrient and drugs. Because of the reason we are looking at these portions, but the other factor is the kidney is basically receiving a lot of high pressure, which facilitate the drug accumulation as well. So I think 2 days ago, the advocacy group from University of Washington already nicely walked us through this system. We employed the notice tip, and basically exact the same one we use. Briefly, we have the flow of the media flow in the strip, and this pink part we can see the cells to get the cells to form the tubular structure. These are just example of the confocal imaging with and without. Oops, that's too fast. With and without drug, so that nicely capturing these morphology of these tubules. Then, as I mentioned, I'm an end user, but so notice nicely already validated the feature of these transporters in these tubules. Both base lateral side and apical side key transporters, for instance, OAT or CD2, with the conformation of the functions using the substrate and inhibitor combination. And also, they confirm the expression of the megalin, which is important for endocytic function for this apical side uptake. So now, using this system, I think this is kind of the main reason why we select for this model for our for assessment of the drug-induced nephrotoxicity. So now, my time validation of this chip. I just provided some example. This is our standard timeline for the drug-induced nephrotoxicity assessment. Notice kindly precede the cells and provide us the chip by delivery. We culture a couple of days or 1 week, and when we start compound exposure, keep culturing for a week. That is general, but you can extend up to 1-2 months as you need. And during this week cultivation, we collected the effluent for a couple of time points for the biomarker production assessment, and end up with the cell viability usually. But you can apply this for the immunofluorescence, RNA collection, or whatever you need. And this is example, many papers already published with the polymyxin B toxicity, so not necessary to explain, but initially you see this kind of the cell swelling, and followed by the massive damage in dose and concentration and time-dependent manner. And by you applying with the pressed blue reagent, you can measure the cell viability. This is the two identical experimental result showing the pretty much nice reproducibility to calculate the IC50 value. Now this is a little bit busy slide with these a lot of graph of the biomarker production of the kidney safety marker and the pro-inflammatory marker. And you can see this nice reaction of these cells to produce those biomarker productions. For instance, high concentration of polymyxin B, which is marked as red, you see the quick upregulation, production of the KIM1 and quickly go down because cells die. This pattern pretty much reflected by these other biomarkers such as clathrin. And if you see the TNF-alpha, also pretty much the same pattern. Middle dose, for instance, orange 50 micromolar, following this pattern of the 100 micromolar, 25 micromolar, which is the middle one, you see the gradual increase of the biomarker, including clathrin and the TIMP1, which is maybe minor. I think no one used this one, but we found that the TIMP1 may be useful biomarker as well. And other pro-inflammatory cytokine, the IL-6, clearly showing this nice correlation with the other biomarker as well, and CCR2 as well. So this is just example with polymyxin B. Now another super busy slide. You don't necessarily to read it, but this is a summary, busy but summary table of the biomarker production for each class of the nephrotoxicant. And for instance, the cyclic lipodecapeptide such as polymyxin B, you see pretty much every biomarker production by those compounds. If you look at the aminoglycoside, which is nephrotoxicant from the apical side uptake usually, we really don't see anything at the safety marker. Maybe only the clathrin momentary production. But you see the clearly nice the production of the IL-6 productions. And other example is macrocyclic, like cyclosporin A. You don't see the KIM1 expression, but you see the clathrin and the TIMP1 production as well. And maybe, but not much with these inflammatory cytokine productions. Negative control, you don't see anything, basically. Then tenophobia, which is base lateral side uptake nephrotoxicant. This is but the apical side, so we don't see. We don't have these full data set though, but basically we don't see any production of that. So now switch gear a little bit about my talk is with this in-house molecule example. Once again, GNA1 experienced this dose-limiting toxicity. With these nice retention of the kidney. And when applied to the human chip, you see these are similar to the polymyxin B. You see the concentration and time-dependent damage of the human in vitro proximal tubes. And this time, we also check the viability. You see the nice IC50 calculation in 3D system, which is clearly left shift as compared to the 2D system. So demonstrating the good sensitivity to capture the toxicity of the compound. Then this time, we show you this rat model, which is we use the SD rat primary RP tech cultured in the same system. You see the pretty much same trend, but with any reason we don't know exactly what happening though. But the rat system actually less sensitive to the compound. So we worried about that, and polymyxin B as well. You cannot kill this 100 micromolar. You cannot kill this rat chip with 100 micromolar. But by applying with tunnel assay, you see the clear signal of this DNA fragmentation, demonstrating that even in that system we see this cell test by the compound, which is exactly we wanted to see because we saw in the preclinical species. I mean the in vivo study. So from few minutes from now, I will show a couple of examples, the capability, what we can do in this system, the mostly confocal microscopy basic assessment for future consideration in the pathology space. Then this is a uptake of the compound of GN2 is the body B fluorescence labeled compound. Then you can see this a nice uptake of that compound by the confocal microscopy. Then this is simple immunofluorescence to against the LAMP1 and the HO, which is oxidative marker stress by the gentamicin and the GNE1, for instance. You see the nice accumulation of the LAMP1 surrounding the nuclei, also the expression of HO1. And this is another example, maybe jump from the so far. We are talking about biphosphonate is a medication for the osteoporosis in the both rat and the human system to look at the mitochondrial activity with the TMRM, which is a dye to capture the mitochondrial membrane potential. So that means if you lose a signal, meaning a reduction of the mitochondrial activity, then as expected, we will see the nice reduction, I mean the decrease in the signal by the compound and the polymyxin B as well in both human and the rat system. I think this is my last data slide, maybe highlighted for the pathological use. We wanted to apply this system for the EM-based observation because of this kind of the pretty much standard in vivo study to assess the compound-induced toxicity. This is on data images using the healthy human in vitro proximal tubule, and but technical wise we can do that. We can we can see the nicely process the samples. Then you see the brush border, the density of the brush border different, totally different from the in vivo situation. So, but this is kind of the further discussion how we can improve or we can make other cells to get this kind of morphology. But bottom line, we see the brush border, we see the mitochondrial organization, and probably vesicle may be formed once we apply this system with the compound exposure. Then we see this border of the cells as well, so probably we haven't stained with the ZO1, but actually notice, I believe notice did the ZO1 staining, so it is clear that we have the title junction as well. So with that, so this model can capture these pretty much nicely the compound-induced toxicity when compared to the 2D system due to these probably enhanced transporters and other endocytic receptors as well. So that we can use that system for assessing these compound-induced toxicity for these even in-house compound by using these a series of the readout, especially we are pretty much pushing using these viability assay and biomarker-based assessment. But that not limited to, we can do any imaging-based assessment for whatever the reason you have. And we can do the same experiment using the LAT system as well, but actually there are a lot of challenge in LAT systems so far we so far experienced because of that some sort of the difficulty in maintaining the culture for a long while with the LAT system. So this is another discussion point in the future for use. Yes. All right. With that, I'm closing my talk by acknowledging a lot of people. Once again, there are many members involved in our work, but especially Vincent, who works to generate the majority of the data set, run and validate in the LAT system for the in-house molecule. Leslie work on the cross-species comparison in this system using reverse phosphonate. Thank you so much. With that, thank you so much. I will take any question you may have.
Yeah, many thanks, Tomo, for presenting your data. And I already see there's one question.
Yeah, very nice talk. I was curious, some of the ranges of the IC50s you were showing were tens to hundreds of micromolar. How physiologically relevant are those concentrations?
Usually, that assessment is that value should be used for the comparison between the exposure level. Then we can do this some sort of the exercise for the, using the margin of safety. In this case, it's a lot of question I have as well. Because of that concentration in system we apply is basically apical side exposure. Then, as you know, Dan presented that concentration in the urine system may be concentrated. And we usually use this safety margin of safety based assessment using the plasma side concentration. So a lot of dissociation between that. So maybe short answer is we don't know yet.
Many thanks, Tomo.
Many thanks.